Welcome back, ladies and gentlemen. So, last time we were talking about mechanisms for which eukaryotic cells are capable of controlling transcription. So, that process of going from DNA to RNA. When we look at prokaryotes, that really is the only level at which they are capable of controlling their gene expression. In eukaryotes, though, we can control it at the chromatin structure, we can control it at the transcription level. We are also capable of controlling it post-transcription. So these regulatory mechanisms can operate at various stages after transcription because there are multiple steps. When we look at protein synthesis in prokaryotes, almost immediately as that messenger RNA is being created, it is already being translated into protein. That's not the case in eukaryotic cells because that nucleus is in the way. So they have to finish transcription before they can start translation. And as a result, that means that eukaryotic cells have much more opportunity to finely tune their gene expression rapidly in response to environmental changes. So we're going to talk about some of these mechanisms here in this video. We're going to talk about RNA processing. We're going to talk about messenger RNA degradation, the initiation of translation, as well as protein processing and degradation, all of which are steps within the protein production pathway post-messenger RNA creation that eukaryotic cells are able to attach control systems to. So when we look at RNA processing, remember that we talked about this idea of alternative RNA splicing. So initially, as soon as that messenger RNA is completed at the end of transcription, it really isn't the final molecule that will ultimately get translated when we get to the ribosomes. We have to add that 5' cap, that poly A tail. We also have the introns and exons that need to be spliced up and ligated back together. So remember, those exons are the expressed parts. Those are the components that will ultimately be containing information to make amino acid sequences. The introns are those interrupting sequences that have to get removed. So when we go through that RNA splicing process, we don't necessarily need to take every single exon that was actually part of the RNA coding region in DNA and put it back together. So we can take some of those exons, put them back together, take them and put them back together in different orders. Really just gives eukaryotic cells the ability to make similar proteins, not necessarily identical ones, from the same basic building blocks in the gene itself. So I can have a single gene in my DNA that can maybe produce me 3, 4, 5 different similar proteins, as opposed to needing a single gene for all 5 of those proteins. So it's a much more efficient use of genetic information and space to store that information. And that's what we're looking at here. So in this particular example, this tropin T gene, I've got 5 different exons within that RNA coding region. 1, 2, and 5 are required for the protein's function, but 3 and 4 can be spliced in alternatively and make slightly different proteins at the end of the process. So if I splice together 1, 2, 3, and 5, I get one mRNA molecule that can then be utilized to make a certain protein. If I splice together 1, 2, 4, and 5, I get a slightly different mRNA molecule, which means I'm going to produce a slightly different protein. And so it gives us some additional mechanisms to control gene expression based on what is available in the environment. We can also talk messenger RNA degradation, so breakdown of messenger RNA. When we produce messenger RNA from transcription, I don't necessarily want that molecule hanging around in my cytoplasm forever, constantly attaching to ribosomes and being translated. So the lifespan of those messenger RNA molecules in the cytoplasm is a key determining factor in the amount of protein that will ultimately be produced from that sequence. Eukaryotic messenger RNAs tend to have longer lives than prokaryotic ones. Prokaryotic ones, remember, that messenger RNA pretty much gets transcribed and translated almost simultaneously and doesn't seem to hang out in prokaryotic cells for very long. Eukaryotic cells, on the other hand, their messenger RNAs can hang around for hours, for days, for weeks even, depending on what that message is, what the protein is that they produce, and what are the homeostatic mechanisms going on in that cell at the time and the environmental influences on the outside of that cell. So when we look at messenger RNA sequences, the lifespan is actually determined by part of the sequence that is in that untranslated region at the 3' end, what we call the 3' UTR, UTR standing for untranslated region. So we can utilize those sequences, your cells molecularly can utilize those sequences to determine how long that messenger RNA molecule typically is necessary, probably dating back to evolutionary history of what that messenger RNA codes for and the protein that it ultimately produces. How often do we need that in our cells? How important is it to have for long durations as opposed to just short stints based on typical environmental conditions? So, not only can we just determine how long that messenger RNA hangs out, but we can also regulate the initiation of the translation itself. So initiation of translation of messenger RNAs can actually be blocked by regulatory proteins that bind to sequences that might be in those UTR regions or other structures of the messenger RNA itself. Alternatively, the translation of all messenger RNAs in a cell can be regulated simultaneously. So I can target certain mRNAs and block them with regulatory proteins, or it's also possible to regulate all messenger RNAs at once. And a great example of that sort of all regulation actually happens by a translation initiation factor that simultaneously activates in an egg following fertilization. So once that sperm gets into the egg and completes that fertilization process, the mRNAs that are in that now fertilized egg all are simultaneously activated to work in that now fertilized egg cell to start stimulating things like cell division so that we get that fertilized egg, that zygote, to now start going through division and starting to produce an embryo. So one of the ways that we can control a lot of this is actually by non-coding RNAs. So we spend so much time talking about RNA molecules when we teach this to you, especially as underclassmen, in terms of messenger RNA, transfer RNA, ribosomal RNA. And that's pretty much where we leave it. But that being said, they are not the only types of RNA molecules that exist. They are just the ones that are directly related to the translation and transcription processes, the actual protein processes. We have a lot of other non-coding RNAs that come in as secondary players to control that gene expression. So only a really small fraction of DNA is actually protein coding, or even RNA or tRNA coding. A huge amount of what we used to think was sort of this junk DNA, we just didn't know what it did. It didn't seem to directly correspond to any sort of product. We are now learning that a significant amount of that quote unquote junk DNA in our human genome actually seems to be connected to the transcription of these non coding RNA molecules. And those non coding RNAs are incredibly important to help regulate gene expression, specifically at two places. At the actual messenger RNA translation itself, they also play a role in the chromatin configuration that we talked about before in terms of adding and removing methyl groups, acetyl groups, so on and so forth. So one type of this non-coding RNA are these things called microRNAs or miRNAs. And these guys are small single-stranded RNA molecules, and they will actually do this sort of hairpin looping on themselves to create these sort of two-stranded RNA complexes. And these microRNAs, then we take that actual coding region that we need from that, attach it to a protein complex, and then these guys will actually help to degrade very specific sequences of messenger RNA and end up blocking the translation process as a result. So these little hairpin loops here, the actual microRNAs will hydrogen bond to themselves. We'll use a dicer protein that will kind of clip these loops off. This top part, this gray piece of that microRNA that doesn't have the actual complementary sequence will get degraded, and then that microRNA will be connected to this protein complex. It's basically an enzyme. But what that ends up doing then is that this sequence of microRNA is very specifically complementary to the messenger RNA code, or at least part of it that we want to stop the translation of. So those guys will either directly connect to that messenger RNA and just break it down entirely, or in other cases, if it's already sort of started being attached to a ribosome, that protein itself can basically act as a block and not allow that messenger RNA additional access to the ribosome, which means the translation process also gets stopped. So we can look at those microRNAs. We can also talk about small interfering RNAs, or siRNAs, which are part of a process called RNA interference, or RNAi. Both these siRNAs as well as those microRNAs, or miRNAs, are similar, but the form from which they are created, their RNA precursors, are a little bit different. So while the microRNAs are going to work directly with messenger RNA degradation or stopping the ability for translation to happen simply by blocking ribosomal access to that messenger RNA, when we look at these siRNAs, these guys are playing a much larger role in the heterochromatin formation, right? Go back to the condensing or relaxing of your chromatin structure. Do I have access to that gene because it's loosely wound around those histones? Or have I caused those histones to stick together and condensed that version or that component of our DNA molecule, in which case I won't have access to it? So these siRNAs are playing a much larger role in blocking large regions of the chromosome, causing the condensing of that region of the chromosome to occur. Now, some small RNAs can also block transcription of very specific genes, as opposed to being just big, large-scale blockages as well.
Scientists have been making rapid progress in understanding RNA interference, or RNAi. Many organisms use RNAi to control genes, and it can also be used as a tool in the laboratory, and in the future perhaps as a therapy. This animation will introduce you to the principles of RNAi involving two important types of RNA molecule: small interfering RNAs and microRNAs. Eukaryotic cells have many sophisticated ways of controlling gene expression. In the complex environment of a cell, these mechanisms need to be precisely targeted. There's a group of mechanisms that use small RNA molecules to direct gene silencing. This is called RNAi. Inside the nucleus, most genes that encode proteins are transcribed by RNA polymerase II. The primary RNA transcript is processed by splicing and forms a mature messenger RNA, sometimes called mRNA. The messenger RNA is then exported from the nucleus into the cytoplasm. Here, ribosomes catalyze translation of the messenger RNA to form polypeptide chains that fold into proteins. But this is also where some small RNA molecules can have their silencing effects. There are several types of regulatory small RNA. Small interfering RNAs, known as siRNAs, are derived from longer double-stranded RNAs that are either produced in the cell itself or are delivered into cells experimentally. The introduction of siRNAs or double-stranded RNA is widely used to manipulate gene expression. MicroRNAs are another type of small RNA. Most microRNAs come from RNAs that are transcribed in the nucleus, which then fold and are processed before being exported into the cytoplasm as double-stranded precursor microRNAs. The double-stranded precursors of microRNAs and siRNAs bind to DICER, which is an endonuclease protein that cuts the RNA into short segments. Most siRNAs and microRNAs are approximately 21 nucleotides long. The short double-stranded RNA then binds an Argonaute protein. One strand of the RNA is selected and remains bound to Argonaute. This is called the guide strand. The combination of the RNA and Argonaute, along with other proteins, is called the RNA-induced silencing complex, or RISC. siRNAs direct RISC to bind to specific messenger RNAs. The targeting is precise because it's determined by base pairing between the siRNA and the target messenger RNA. siRNAs often have perfect complementarity to their target sites. Once bound, Argonaute catalyzes cleavage of the messenger RNA, which will then be degraded. MicroRNAs also guide RISC to messenger RNAs. Usually only part of a microRNA, known as the seed, pairs with a target messenger RNA. This imprecise matching allows microRNAs to target hundreds of endogenous messenger RNAs. Targeting by a microRNA can lead to messenger RNAs being degraded or translation being inhibited. Argonautes and their small regulatory RNA cofactors are found in plants, animals, fungi, and some bacteria, and their importance in a multitude of biological processes and as tools continues to be revealed.
All right, so finally, we can also regulate the gene expression even after the protein is produced. So when we look at proteins that get produced from translation, as soon as they fall off of that ribosome, some of them are just active and they can go do what they need to do, but a lot of them often end up needing some additional processing. And so we can utilize that processing process as a way to either cleave that protein in certain places, add additional groups to it, all of which are subject to control based on the availability of enzymes that need to be available for that cleavage to occur, or to add those groups. We also have these things called proteasomes, and they are in themselves giant protein complexes that will specifically bind to protein molecules that we no longer want in our cells, we no longer need based on environmental conditions, and will actually degrade them. So sort of think of it like sticking something into a blender, and that protein kind of acts like that blender and just chops it up into a bunch of pieces that we can then recycle and reuse.